All right, today we're doing something pretty amazing. We're going to decode the instruction manual for life itself. We're diving deep into bioinformation, this incredible field that uses some serious computing power to read, understand, and yeah, even rewrite the genetic code that makes you, well, you.
So here's the plan. First up, we'll tackle the big one. What even is bioinformatics? Then we'll get into why it was so desperately needed. We're talking about a biological data explosion. After that, we'll look at how it actually creates this digital map of life, see how it's popping up in your world in some surprising ways, and finally, we'll make it super personal, looking at how it's decoding diseases to design cures made just for you.
Okay, let's jump right in. To really get a handle on what this is all about, we're going to start with a really simple but super powerful idea that kind of grounds everything else. You know, at its very heart, the name really says it all. You've got bio for life science and informatics for information science. So put them together, and what do you get? It's literally where biology meets the algorithm. And if you want a more, textbook definition, it's this mashup, this fusion of biology, computer science, and IT. But it wasn't created just for fun. It was born out of necessity, for one critical purpose: to finally manage and make sense of the absolutely insane amounts of biological data we can now collect. So if there's one thing to remember, it's this. Think of bioinformatics as the entire digital backbone for modern biology. It's like the librarian, the translator, and the Google search engine for the code that defines who we are. It's the whole shebang. And that, of course, brings up a huge question. Why? Why did we need to invent a whole new science for this? Well, the answer comes down to a problem of scale, a problem of just having way too much information. And this slide, wow, this really tells the story perfectly. On the left, you've got the old way of doing things, that nice, neat little loop of scientific discovery. But then, boom, on the right, you see what happened when things like the Human Genome Project came along. It created this data flood, a massive bottleneck. All of a sudden, we could churn out biological data way faster than we could ever hope to analyze it. And the scale here is just mind-boggling. We're talking over 100 billion, with a B, base pair sequences. Those are the individual letters in our DNA, and that's from hundreds of thousands of different species. To put that in perspective, the very first human genome cost billions of dollars to sequence. Today, you can get it done for under $1,000. That crazy drop in cost created a tidal wave of data. Trying to analyze this by hand? Forget it. That'd be like trying to read every single book in every library all over the world at the same time. Totally impossible. So to handle this tidal wave, bioinformaticians had to build a kind of digital toolbox, a way to organize all these different layers of biological info so they could start building a complete, coherent map of life. And these are the tools, the raw materials in that box. It all starts with genomics. That's the raw DNA code, the master blueprint. Then you have transcriptomics, which is like looking at the RNA messages, kind of like photocopies of specific instructions from that blueprint. After that is proteomics, which studies the actual protein machines that those instructions build. Then there's epigenomics. Think of these as the dimmer switches that turn genes on and off. And finally, metabolomics, which analyzes all the chemical footprints that get left behind by all this activity. And this is where it all comes together beautifully. This image shows exactly how scientists use that whole omics toolbox to build a complete roadmap. You start with the tiny letters of the DNA sequence, see how they build molecules and cells, and you scale it all the way up until you can understand a whole living organism. It's incredible. Okay, so that's the theory. But let's bring this down to earth. Let's see how all this moves from being just abstract data in a lab to real-world applications that are changing our lives, probably in ways you don't even realize. I mean, the impact is just unbelievably broad. Obviously, it's turning medicine on its head, but it's also changing the game in forensic science, in agriculture, and how we protect the environment. It's touching pretty much the entire field of biotechnology. To make this really hit home, let's zoom in on one of the most fascinating areas: how bioinformatics is actually being used to solve crimes. And believe me, this is not science fiction. Of course, you have your standard DNA fingerprinting, but now we're talking about analyzing these tiny single-letter typos in our DNA, they're called SNPs, to actually predict what a person might look like. We're talking hair color, eye color. It's amazing. There are even wild techniques to figure out how old a wound is just by looking at which genes are switched on or off.
All right, now let's shift gears completely. Let's go from the crime lab to the farm field because bioinformatics is also playing a huge role in one of humanity's biggest challenges, feeding the planet.
You see, by analyzing the genomes of plants, scientists can go in and edit their DNA to make them better, improving crop yields, making them resistant to diseases. And it's not just about the plants themselves. By studying the DNA of all the tiny microbes in the soil, we can actually use them to help clean up nasty environmental pollutants. It's a total game changer.
And that brings us to what might be the most personal and honestly, the most powerful application of all, using bioinformatics to understand our own health and to design the future of medicine. So this is the central question, and it's a really profound one. Think about it. Our genetic code is over 3 billion letters long. So how on earth can these tiny, almost invisible variations in that code have such massive consequences for our health? Well, this table gives us a few crystal clear examples. We now know that specific typos or mutations in genes called BRCA1 and BRCA2 are directly linked to a much higher risk of breast cancer, or that a certain version of the APOE gene is a major risk factor for Alzheimer's. Bioinformatics is the tool, the superpowered magnifying glass that lets us find these critical connections hidden in all that genetic noise. But here's the thing. It's not just about the static DNA code we're born with. That's just the blueprint. We can also find clues in the dynamic, real-time chemical reactions happening in our bodies every single second. So by analyzing the chemical fingerprints our bodies leave behind, what's called our metabolite profile, we can actually spot the signs of disease. For instance, an increase in a chemical called lactate can be a marker for cancer. Elevated glucose, that's a classic sign of diabetes. It's like having a biological early warning system built right in. And all of this is leading to the holy grail, really. A new era of medicine called pharmacogenomics. It's all about getting away from one-size-fits-all treatments. For example, did you know that the right dose of a really common blood thinner can be 10 times different from person to person? And it all comes down to a couple of gene variations. Pharmacogenomics lets us predict that. It's about finally being able to design treatments tailored specifically to your unique genetic profile. The right drug for the right person at the right time. That's the dream. So think about where we've come from. We've gone from being completely unable to read the book of life to having the power to search it, to analyze it, and now even to edit it. These capabilities are growing at an exponential rate. So the final question, the really big one, isn't about what we can do anymore. It's about what we should do with this incredible, almost godlike power. And that's a question for all of us to think about.